Morning, everyone. Thank you for joining Speed Rounds. My name is Madhu Anand, and I am with the Regional Epi and Investigations Program in the New York State Department of Health Bureau of Communicable Disease Control. I will be presenting along with Amy Robbins this morning, an epidemiologist in the Bureau of Communicable Disease Control as well. And we'll be talking about the use of whole genome sequencing during foodborne outbreak investigations. So just to give you an idea of what we're going to talk about, we're going to give you a little bit on background and then talk about how pulsed field gel electrophoresis and whole genome sequencing are conducted in New York State, and then specifically some background about sugar toxin producing E. coli. We're going to present a case study to illustrate some of the interesting findings we're starting to see conducting whole genome sequencing with E. coli, and present some challenges not only on that case study, but overall with whole genome sequencing. So to start off, to make sure everyone understands pulsed field gel electrophoresis, this is a method that is used to determine whether two patient specimens might be related to each other. And so what the lab does is they conduct this lab technique. They put on each column a case specimen. The yellow that you see here is basically to make sure that the gel is running correctly. And then when they're getting ready to start the gel, they run an electric current through the gel, and the case patient specimen's DNA bands fall out essentially on a different weight. And so you can see in the area that's white here that those bands match up and would be considered matching. In the red area, specifically in the circle, you can see the bands are not lined up and they are considered not matching. So one of the things looking at PFGE is it's only looking at certain bands and certain segments of DNA. However, with whole genome sequencing, they use inexpensive machines that can now sequence the whole genome of a bacterium. So rather than looking at specific portions of the DNA, we're really looking at the whole matrix. And so behind the machines, there are computer programs that exist that can compare these genomes to detect what's called single nucleotide polymorphisms, or SNPs. In this example, we have two bacteria. In essence, you can even call them two individuals, even if you wanted to say that. And we basically put the whole code, the ACTG code, in line for that specific bacteria or individual. And they compare against the two, and they look and see where there might be a difference. So where this A and T are, there's a difference here with a C and G, and it's considered a SNP difference. So those SNP differences then get illustrated onto a phylogenetic tree. This is an example of how that tree would look like. I am not going to go into lots of detail on the tree, but essentially I wanted everyone to know that these letters here would represent a different organism or a different person. And basically that if they're connected all the way to the left, that means that there's a common ancestor at some point, and they've diverged at certain points and gone through differences. How different they are measured by what's called SNPs, and that those SNPs and this tree can reveal genetic closeness and ancestral relationships. One of the things you'll hear a little bit about as we continue talking today is at what level is a single nucleotide polymorphism relevant? Is it 5 SNPs, 10 SNPs, or more? And we'll talk a little bit more about that. So to talk a little bit about the process of PFGE and whole genome sequencing in New York State, I'll talk first about how PFGE is used to conduct pathogen-specific surveillance for outbreaks. So a commercial lab tests specimens submitted by providers, and they send those isolates to Wadsworth Laboratory. Wadsworth then conducts pulsed field gel electrophoresis and uploads to a national database called PulseNet. And then all states are uploading into PulseNet at the same time. And Wadsworth reviews the information in PulseNet to see whether or not there are other DNA matches. They then send those matches to the New York State Bureau of Communicable Disease Control, where the FDAs there will review the information that local health departments put in the Communicable Disease Electronic Surveillance System, or CDES. They will conduct follow-up if needed by reviewing information, summarizing the information in databases here, and then reaching out if additional information is needed. They share with partners, both internal and external, this information, the summary information. They try to identify links and maybe conduct traceback, conduct inspections, or collect samples. Now that whole genome sequencing is being introduced, the workflow that is occurring at Wadsworth is changing a little bit. So in day 0, when a bacterial isolate is received at the PFGE lab, it takes about 2 days on average for PFGE to be completed. The same process of PFGE results being uploaded to PulseNet occurs, and those matches are reported to EPIs. We then start our investigation. Subsequent to the PFGE being completed, what's happening in parallel at that point is whole genome sequencing is occurring. And it can take up to a week after PFGE is completed for us to have a whole genome sequence. That whole genome sequence data is shared with federal and state collaborators such as CDC. That data is then analyzed. It can be analyzed in 2 different ways. It can be analyzed at CDC, but we also at Wadsworth have an in-house analysis as well that's completed. So that analysis basically tells us, okay, are there any matches? And those analyses are reported back to epidemiology as well, where we can then refine our investigation and determine whether some cases might fall out or in of our investigation. So right now, what foodborne pathogens are routinely submitted for whole genome sequencing? Right now, Wadsworth is conducting on whole genome sequencing on all isolates of S-Tech Nano 157, Listeria, and Salmonella. However, on E. coli 0157, whole genome sequencing is just conducted by request of the Bureau of Communicable Disease Control. So as far as background E. coli goes, Shiga toxin-producing E. coli are caused by a toxin-producing bacteria. And basically what happens is you ingest the bug is in your gut and is proliferating toxin and is making you sick. The way that you become sick, the symptoms and complications that are seen traditionally are stomach cramps, diarrhea, often bloody diarrhea, vomiting, and hemolytic uremic syndrome, or HUS. And this can be pretty serious. Hemolytic uremic syndrome is basically a complication of the kidneys. It impacts children more often, and often you're put on dialysis if you have HUS. The incubation period is about 1 to 10 days, but usually it's between 3 and 4, and the duration of illness is 5 to 7 days. There are 2 main subtypes of E. coli, 0157 and non-0157, as I've mentioned. And in recent years, predominantly due to changes in testing methodology, we've been able to identify that a majority of the E. coli we're seeing out there is E. coli non-0157. I think this slide says 65 to 35%, but I think it's like getting closer to like 70-30 even. So there are approximately 7,000 cases reported in the United States each year, of which about 200 are reported from New York State. And as you can see, our numbers have stayed relatively steady in New York State, excluding New York City for the past several years. We do hover around that 200 mark, maybe a little bit more, maybe a little bit less. This slide has broken out into E. coli 0157 and non-0157, reflecting that E. coli non-0157 is being seen more. The other interesting thing is, as time has gone on, our E. coli non-serotypable has decreased to pretty negligible amounts as well over time. So I think one of the things that is important to talk about is, why are we specifically interested in whole genome sequencing for E. coli? One, as I mentioned, it's a very severe disease, and so we do want to investigate cases of E. coli very intensively. And even though we are better able to determine E. coli non-0157 cases now, finding the specific serogroup of non-0157 is harder to identify at a local laboratory. So having whole genome sequencing available more quickly at Wadsworth is able to help us find information on a serogroup and help with an investigation more quickly. In addition, recent investigations have indicated that whole genome sequencing may better identify a cluster. We've had a couple of instances where we've had, even in the same investigation, where we've had multiple PFG patterns found to be related by whole genome sequencing. So our case definition for what we're including as far as cases has to be adapted. And likewise, we found that the same PFG pattern that we thought was maybe one cluster is now split between different whole genome sequencing clusters. So has helped us refine our investigations as well. So that being said, I'm going to switch off to Amy Robbins, who can walk you through a case study.
So the case study we're going to use occurred about a year ago, and this was a cluster of E. coli 0121 Shiga 2 cases. We were first notified of these cases on January 28th from the Wadsworth Center, and they had identified a PFGE pattern that matched 8 cases nationally from 7 states in the past 60 days. So on the next day, CDC noticed that this PFGE pattern was above baseline for them, and so they assigned it a cluster code, and they included 12 cases. The ages were between 13 to 70, and 79% of these cases were female. The New York State case that was included was a 70-year-old female with onset of illness on January 12th of 2016. She had bloody diarrhea, dehydration, weakness, and hypoglycemia. She was hospitalized for her illness, and she resided in a county in central New York. So based on the national cases, CDC decided, getting ahead of myself, but we here in New York State use a hypothesis-generating questionnaire we developed. It's an 11-page questionnaire. It takes about 30 to 45 minutes to interview someone with this questionnaire. And it asks a lot more detailed information than our initial questionnaire does. And it asks questions about meat exposures, quite a few about ground beef, about vegetables, fruits, animal exposures, as well as about grocery stores and any places that they might have consumed food outside of the home, whether it's a restaurant or an event. So our case had quite a few fish, seafood, and meat products, as well as ground beef, quite a few vegetables that were all consumed raw, as well as some fruits, and had exposure to a cat and a dog. Our case shopped at Wegmans, which has shopper cards, so we did ask permission and received written approval from her to use her shopper card and gain her history of all the foods that she had purchased at Wegmans about 3 months before her onset of illness. And she'd also attended an Elks Club event. So for the national investigations, we share data with CDC about our cases, and each state shares this information. We have a system that's available online that's a protected system, and the states can all input all their exposure information for each of the isolates that match a national cluster. And what CDC found was, of those 12 cases that were initially part of this cluster, common exposures between those cases were leafy greens, ground beef, and other vegetables. So CDC developed an outbreak-specific questionnaire that looked at more detailed information about leafy green consumption, ground beef exposure, and other vegetables. So our student interview team, known as Team Cluster Buster, interviewed the New York State case, and we shared all that data with CDC. While all these interviews are happening, the Wadsworth Center is taking the isolate that they found and doing whole genome sequencing on it. They're sharing this data with CDC. A couple of other states are also doing this at the same time, as well as CDC, and they're finding that the New York State case and 3 other cases nationally were considered indistinguishable by whole genome sequencing. So this is an example of the results we get back from CDC. As Madhu mentioned earlier, this is called a phylogenetic tree, and it shows that these 4 cases here all match and are considered indistinguishable with a 0 SNP difference. The third one down with the arrow is the New York State case. To compare it to another cluster of cases that was occurring at the same time but had a different PFGE pattern, so this here is the different PFGE pattern, they put these 2 cases on the same tree to show that these are not related because they are 77 to 79 SNPs different. These 2 cases down here are both New York State cases that also occurred from a central New York county around that same time period. But based on whole genome sequencing, they would not be considered part of this cluster. So the results of the outbreak-specific questionnaire show that there was no statistically significant food item amongst those cases, and additional cases were seen nationally. So CDC decided to go another step to open-ended questionnaires. And these are interviews that are very extensive. They're about an hour or longer. And this is where there's one interview at CDC who interviews all the cases across the country, typically the newest cases part of the cluster, and asks a lot of questions about food consumption, activities related to food, animal exposures, travel history, just a lot of questions. And through those open-ended questionnaires, of which the New York State case was not part of this, they did identify that a lot of the cases had done some type of baking during their exposure period. So this tree here just shows, as we're continuing with the investigation, we have a lot more cases that are 0 to 1 SNP difference, which means all these cases most likely had the exact same exposure, and these cases were throughout the country. So now, based off of the open-ended questionnaires, we have a second outbreak-specific questionnaire, and this one focuses specifically on baking and baking products. So it's asking questions about consumption of raw batter, raw cookie dough, what type of baking soda is used, what type of flour is used, what type of chocolate chips or anything else, nuts that are added to baking. It was about, I think, 5 pages long, this questionnaire. But what they found out is that to do statistical analysis, they have to compare to consumption in the general public. And so all the information that we have about general public consumption is about meat, about fruits, about vegetables, but there's no questions on baking exposure. There's no questions about flour. So we had nothing to compare to see, is this statistically significant, this baking exposure, or not? So CDC decided to do a case-case comparison study. So this is taking for each of the cases that occurred as part of the cluster, they're taking and they're matching 4 controls to find out about baking exposures. To do this, instead of doing the more traditional random digit dialing of trying to find somebody in that geographic area, they matched by state. So for our case in New York State, who was a 70-year-old female, we had to match it to 4 controls of a non-E. coli enteric illness. So we used Campylobacter and Salmonella cases that were reported to the states during this time frame that we were doing the interviews, which was in April of 2016, that were over the age of 30 and that were female. So in a week period of time, we did have 4 interviews conducted with a Campylobacter or Salmonella case, and local health departments were notified of cases that came in and were reported and asked if they could complete this questionnaire when they did the initial interview for the Salmonella or the Campylobacter case. So while that was all happening and we were doing the case-to-case study comparison, a lot of the states had found that their cases had some of the products available that they'd used in baking. And not only did they have the product available in their house, such as the flour, they also had the original packaging. So the original packaging for a number of the flours was collected as well as some samples. Some isolates were found of E. coli 0121, and PFGE showed it was the same pattern, and then whole genome sequencing showed that the flower was also 0 to 2 SNPs. So what you see here that are in the 0 to 2 SNP range are either stool specimens from cases or flower isolates that were from the flower samples. So as a result of the whole genome sequencing showing that the flower and the stool specimens matched and were considered indistinguishable, and the information that came from the EPI study of looking at information about flower exposures, General Mills did issue a recall in May, and then they issued a second recall in July. And this is an example of what's still up there on their website based about these recalls. So typically that we would hope that was the end of the story, and that with the recall we didn't see any more cases. Unfortunately, that's not the case for something like flour, because people empty the flour into bins and then they just continue to use it, and they don't realize what type of flour, or don't remember what type of flour they purchased. So in July, we did have another case reported. This would have been our second case. It was a 16-year-old male from central New York County, and the PFGE matched. But this individual did not have any flour exposures identified. In August, we had a third case, and this was a 27-year-old female, also from a central New York County, and then no flour exposure was identified when she was interviewed as well. And these cases were also interviewed with the outbreak-specific flour questionnaire. And then we had a third case after this recall, which was in September, and this is a 24-year-old female who did say that she had raw cookie dough with an unknown brand of flour. So with these three cases, we did whole genome sequencing at Wadsworth, and this is where things get a little complicated and one of the challenges that's coming out of it. So in the right-hand corner, these are the isolates that are part of the cluster by CDC. They have the 1601 MLEA1, which is the outbreak cluster code. We have our very first case, which is the one in the middle, that was reported in January of 2016. And then we found that 2 of those other 3 PFGE patterns were 0 SNPs, different, meaning indistinguishable, and most likely the same common exposure. What we found was that the other case, the third case, which was the one that was reported that had an onset of illness in August, actually did not match by whole genome sequencing, even though it was the same PFGE cluster, PFGE pattern. But it was 64 to 66 SNPs different, so would not be part of the cluster associated with the flour. And this case also did not have exposure to flour identified in any of her interviews. Unfortunately, we found this out after CDC did the final website posting. So if you go and look at it, you'll see that there are 4 cases from New York State included, when really only these 3 cases are matched by whole genome sequencing. Interestingly, at the very first tree I showed, there were those 2 cases that were similar PFGE pattern that CDC had done whole genome sequencing back in January. They'd done the analysis and found that they were not part of the cluster. This case from August matched those 2 cases. And so it was a second cluster in a central New York county, and we never did find out a common exposure. Those 3 cases occurred over the space of a year, and there was no commonality identified. So this is the more complex nature of whole genome sequencing, but also how it's helpful is that it can really limit, even though the PFGE matches, whole genome sequencing shows that it does not. Smithy's going to talk about some other challenges.
So, as I alluded to earlier and illustrated with the case study, we did have multiple PFGE patterns that ended up falling into a single whole genome sequencing cluster associated with flour exposure. At the same time, we also had an E. coli 0121 PFGE pattern, as Amy was indicating, that initially looked like it would have fallen into the baking exposure cluster, but based on whole genome sequencing was separated out and had its own distinct cluster. We also have some challenges with being able to interpret PFG and whole genome sequencing on food samples. When we look at the national database, our federal partners are sequencing on a regular basis, both the Food and Drug Administration as well as the United States Department of Agriculture. And they do upload routine sampling results into this database. Sometimes when we conduct investigations, we see a number of different food items that could possibly be clustered with case patients. And sometimes it's not just all beef. Sometimes it's beef and leafy greens and facility sponges. And so we need to understand better what weight to put on routine sampling results, which could cluster by whole genome sequencing. Furthermore is interpretation. We've had a lot of conversations with Wadsworth in the past several months about how to define a cluster. Wadsworth has actually been conducting whole genome sequencing for a number of years now, and we keep refining how to define a cluster. I think it's still relatively new, and so initially Wadsworth was whole genome sequencing, and a year, 2 years later, if there was a match, that was still getting reported to us from an isolate that was in the past. Now we're starting to talk about specifically for different pathogens, should we be looking at a 60-day window, which is what we do for PFGE for certain pathogens? Should we, for E. coli, be looking at a year? As Amy pointed out, with this E. coli 0121 cluster, we did have cases that spanned several months because the product could have been in bins. And so it was important for us to keep a wider window when looking at E. coli. Number of cases. Sometimes 2 cases are enough to define a cluster for certain pathogens. For Listeria, 2 cases can be quite illustrative. But for something like Salmonella, we probably want to look at more of a threshold of at least 3 cases before defining it as a cluster. We also want to look at those SNP differences I was mentioning earlier. Amy started taking a look at showing you on the phylogenetic tree. It was pretty clear when it was 0 to 2 SNPs that those cases were related to each other. But when you start taking a look, and I think the other really clear part is when there's 70-some SNPs, you have a cluster 70-some SNPs apart, that they're different. But when you start looking at 10 SNPs or 20 SNPs, for some of the Salmonella serotypes and for E. coli and Listeria, you might want to look for surveillance purposes at that area as well. And so when you take a look at time, number of cases, and tightness of cluster, the number of clusters you're investigating can vary greatly based on how you're defining each of these items. So then one of our other really big issues is communication. We have learned that we need to communicate a lot with different partners within New York State. We are having monthly meetings now with Wadsworth to talk about some of these thresholds mentioned on the previous slide and how to define a cluster. But I think one of the other things that we're trying to communicate as well is Wadsworth is trying to relay the information to us. We're trying to investigate, and then it's important for Wadsworth to know what the result of our investigation is so they can refine some of their methodology, and it can also help with providing them some closure on metrics and performance. Likewise, we also like to communicate with our Bureau of Community and Environmental Health and Food Protection. A large part of investigations is that 3-legged stool. It's both the laboratory, epidemiology, and environmental health playing together to identify a source. So they are routine partners at the table when we discuss any cluster. We also need to communicate to local health departments, including New York City, our results and kind of the summaries of our investigations, as well as request follow-up information. It's been a challenge trying to communicate whole genome sequencing. We've gotten a lot of questions about, these gray areas of SNP differences. What does that exactly mean? And it's not easy to, sometimes we don't have the answers. We don't necessarily know what it means, and we're exploring it so it can help inform the future, maybe not necessarily immediately right now, but more down the road. We also communicate with our out-of-state partners on a regular basis, whether that's state or federal partners. We need to know kind of where whole genome sequencing is at with our other partners. And sometimes there are different methodologies used to analyze whole genome sequencing data. So we also have to understand how we're doing things differently. So another aspect of communication is how we're sharing the information. As mentioned, there are different methodologies right now for doing whole genome sequencing, and the nation is now starting to, and CDC is now starting to come to a standard and trying to develop a nomenclature to talk about, to be able to relay information to states, and we can then relay it further to our in-state partners. It's under development, I think, for some pathogens, primarily Listeria. It might get out within the next year. And so in the next year, you might start to be seeing more information from us about that. But then even how to communicate that information. Initially working with Wadsworth, we were communicating information between the lab and Epi via email. That has now transitioned into a SharePoint site where they communicate the lab results to us. We then put some Epi information in and put what the outcome of the investigation is. And as they are able to analyze into phylogenetic trees, those phylogenetic trees are included on the SharePoint site. We are also, from the Bureau of Communicable Disease Control, as mentioned, struggling with how to share some of this information with local health departments. And that is something that we're starting to think about as well. And then I don't want to minimize the resources that are needed to conduct, interpret, and communicate whole genome sequencing. Even though PSGE involves, I think, more skill than running a whole genome sequence on a machine, there is skill involved when it comes to analyzing the data. And we're lucky at Wadsworth because we have a bioinformatician who can do a lot of this. I know a lot of other states struggle with that part of it as well and lean heavily on CDC, but then CDC is getting a lot of requests as well. So there is a priority list. So there is skill involved with analysis. There's also time. Right now, you saw in that initial timeline that it could take up to 10 days to get whole genome sequencing analysis results, depending on the time of year and how many specimens are coming in. That could shorten to, within a week, less than a week to up to 3 weeks, depending on how quickly those specimens can be sequenced. It does take money to get the machines up and running and sequence everything. And then personnel. We've talked just about one case, and there were a lot of different people involved in the investigation, from Wadsworth, Bureau of Communicable Disease Control, Environmental Health, local health departments, CDC, FDA. There's a lot of personnel that go into investigating these whole genome sequencing clusters and trying to put the pieces together. And since whole genome sequencing has the possibility to identify more and more clusters, that personnel and time is just going to increase potentially to investigate this at the same level we have for whole genome sequencing. Some additional considerations as well that we are still trying to get our head around is there is the ability to identify antibiotic resistance through whole genome sequencing. That is something that Wadsworth is starting to take a look at. we're still trying to learn more about that. And the other big consideration is right now, everything that we're doing for foodborne surveillance is very dependent on having an isolate at Wadsworth, specifically for Salmonella, E. coli, and Listeria. In this day and age, culture-independent diagnostic testing is becoming more and more adopted. And we might not always have an isolate that's being submitted to the state public health lab. And so will whole genome sequencing be able to be tested from stool in the future? There are some inherent complications with conducting whole genome sequencing in stool, because what are you finding in the sequence? Is it just incidental, what somebody may have consumed? Is it the pathogen? And so there are a lot of, I think, complications potentially from doing it from stool, and that needs to be researched more. I would like to thank a number of individuals. We could not do what we do without local health departments. You guys are invaluable with the investigative work you've done. Also, my colleagues in the Bureau of Communicable Disease Control, as well as the Bureau of Community Environmental Health and Food Protection, Wadsworth and the Division of EPI, and the guidance from CDC. And we'll leave it open for questions.
Okay. We don't have any questions in the chat.
Okay. So everyone's been unmuted. We'll stick around if you have any questions.
But about the flour issue, is that so? They know where the contamination happened before the flour got, or it was ground and processed, or do they know if it happened afterwards? I mean, with something like this, you're trying to figure out. I mean, it's happening 6 months after the case. And, I guess you're trying to figure out if there's a way, something in the chain to get fixed for future things down the road.
So for people on the phone that might not have heard the question, it was basically what happened with the flour? Why did it get contaminated?
I think that's something they're still looking into. I haven't seen anything come out exactly saying this is how it happened. I know that General Mills is looking into it.
Yes, thank you. They haven't been able to get away.
And CDC has definitely been working with them. And I think that they've been working more, I think, with FDA to try to figure out what is going to USDA folks to figure out where that contamination might have occurred. But I think they did find it from, I think, 2 facilities, and they did do a lot of environmental sampling as well.
And I think one of the really interesting things about this investigation is, in the past, they've suspected flour for a couple of E. coli outbreaks, like 1 or 2 previously, but they've never been able to identify it out of flour. So this was the first investigation where they were able to get it out of the flour.
Because the first one was 2009 with the rock cookie dough that was purchased at the grocery store.
PFG match can also be separate whole genome sequence clusters that you're testing the whole. But how is it that you gave another example where one single whole genome sequence cluster, when you look back, can be 2 different PFG matches? How is it that happens?
So and I am definitely not a laboratorian. I'm going to guess here and say that I know that I've heard that sometimes plasmids can make it look like they're different PFG patterns when they're really the same. So depending on how you're analyzing the data, that could be one area. I think that's one aspect of it. And I think sometimes you're only looking at certain bands, so it might be giving the resolution that you need.
Some of the PFG patterns that have been in the same whole genome sequencing cluster are very close.
Is it the same recipe no matter where you go? So is there consistency across EFGE? So our whole genome sequencing. So is whole genome sequencing done here give you the exact same results that whole genome sequencing done at CDC would?
So there are different machines. So they, I think they would largely give you the same result as far as the sequence goes, but then it's a matter of how you analyze it. So, you know, what regions you're analyzing, what your quality control is, and of the read, which gets into much more technical terms. But I think that's where you start to see differences.
Although with the bioinformatics, I mean, they're standardizing that as much as possible. Even the purchase of the machine, he's been purchasing some for states to use, and they're again purchasing using the same equipment to try to minimize that.
So the comment from the that, you know, one of the things CDC is trying to help with is standardizing it across the nation by helping provide funding for specific types of machines and starting to work at making sure there's standardization with bioinformatics.
Now, the cluster, I mean, the cases all seem to come from central New York. So, were there certain lots for General Mills that have been, that, you know, on the shelf there that weren't anywhere else? Like that,
Yeah, maybe they do more baking. I don't know. There were a number of products that were recalled from General Mills that were sold, I think, at different stores. So I don't think we have that level of information. Maybe David would know that from the environmental health side of things. As to what, but it was kind of interesting that all of our cases were in the same area. But we're also relying on people actually going into their provider, collecting a stool specimen, and so all of that too could limit it. But I know there were just a few cases in each of the states. So there were other states with just one case, or two cases, or three cases. It wasn't like it was really just one state had a ton of cases and the other states didn't have as many.
Now, some of the PFT matches that were not whole genome sequence matches, were they still in the same county?
There were, I think, 4 counties in central New York involved in the 2 clusters.
May also be some questions. Is whole genome sequencing really the whole genome? Or is it basically, you know, is it like from 1 to 10,000, or could it be from, you know, 100 to 9,000 type of thing? And does it matter? So I guess where I'm going with this is there a net, like there is PulseNet, where if everyone kind of puts these sequences in, you have to like start at the beginning and start at the same beginning and start at the same end for all these assorted bugs that you are.
So the first question is whole genome sequencing really sequencing the whole genome? So ideally, yes, but in reality, I mean, it's billions of base pairs. So it's technically not the whole genome, but it's a good portion of it for bacteria or viruses, depending on what you're looking for. What they try to do is look at conserved regions of the genome and do those areas, sequence those areas. And then what was the second part of your question? PulseNet. Is there a national repository where all these genomes reside? And so actually whole genome sequencing isn't all that new. It's been done for years and years and years, just in more research areas. And all those sequences reside on a platform at NCBI, which is, sorry? Oh, so Genome Track. So yes, so GenBank. So it's NCBI is the National Center for Biotechnology Information. And so now what's happening is all these state public health labs that are starting to sequence are uploading, once they have proper quality of their sequence, are uploading into that NCBI database with de-identified data elements. So you would never be able to go back and say, oh, it's from, you know, Lewis County or, Nassau County, that, you know, a 27-year-old female. But it's basically saying you would be able to analyze and come up with a phylogenetic tree and say, oh, these two sequences look like they might match each other, and have some basic background information. So now, so that's where the bioinformatician comes in, because they would be able to go in and grab that data and let you know what the SNP differences are. And that's where CDC is starting to develop some software, so state public health labs can do a similar thing. Any other questions? No. Okay, great. Oh, and if anyone wants the recording, you can either email me or Dwayne Barker, and we can send you the link to the recording. Okay, thank you.